Hello and welcome to my talk. My name is Lukas Krusenbaum. I am a PhD candidate in the phenotyping cluster of excellence, and I am happy to give you an introduction into my ongoing work on rice root imaging and phenotyping for large-scale characterization of rice genetic resources. The goal of this project is to identify novel donors of root architectural traits for rice breeding programs in Madagascar, specifically targeting small-scale farmers and low-input production systems. A large genetic resource of 3,024 diverse rice accessions is publicly available within the 3K Rice Genomes projects, along with genotypic data consisting of single nucleotide polymorphisms for all of these accessions. As phenotyping root architectural traits for all 3,024 accessions is not feasible, the current project is aiming to phenotype a subset of 500 accessions representing more exotic subpopulations of rice in Bonn and Madagascar, and to use the resulting phenotypes to calibrate a genomic prediction model to predict the remaining accessions in the set. The rice root system is very complex with 4 types of root classes. Primary and crown roots form the main root axes and give rise to 2 types of lateral roots. The large lateral roots, or L-types, contain multiple layers of cortex and produce secondary branches. The small, or S-type lateral roots contain a single layer of cortex, are very thin, and only grow to a maximum length of approximately a centimeter. The latter have been linked to efficient phosphate uptake under low phosphate conditions, making these an obvious target for genetic improvements of the rice root system under low input conditions. However, given the variability of abiotic stresses experienced by plants in the fields, different idiotypes may be beneficial for different stress conditions. In our study, we are using 2 different growing conditions for phenotyping of root architectural traits. While rice can be grown in hydroponics, making it easy to study the root architecture, whether phenotypes estimated for accessions under these artificial conditions are also relevant under field conditions remains elusive. We have used disjoint sets of accessions in Bonn and Madagascar to calibrate our genomic prediction model on data acquired in hydroponics under greenhouse conditions, and testing it on a smaller, independent set of accessions grown under upland field conditions in Madagascar. While a lot of software solutions are available for extracting phenotypes from scans of whole root systems, their ability to distinguish the different classes of lateral roots in rice is limited. Also, given the variable developmental status of different root axes, human input is required to ensure that trait values measured on individual roots are comparable across plants and genotypes. Separating the densities and lengths of L-types and S-types on all main root axes has previously been done by manual phenotyping, where human annotators would draw along root axes and count different types of lateral roots in subsections. As this methodology is based on human vision, it produces only small errors. However, major workload bottlenecks of freehand drawing and manual counting make it tedious and time-consuming. Within a collaboration between PhenoRob and the Iowa State University Department of Agronomy, we try to reduce the time spent on these tedious tasks while retaining the concept of basing phenotyping of lateral root traits on human vision to reduce errors. We used existing software packages to generate skeletonized representations of whole rice seedling roots, which can then be used to identify tips and branches of the root system. As the S-type laterals contain only a single layer of cortex, these can be separated from L-types when thresholding root scans based on their grayscale value. By using A-star search, we can find paths through the whole root system, representing the main root axis as the shortest distance through the root system between a root tip and the seed. Once the main root axis is identified, the same principle can be applied to lateral roots by using any point along the main root axis as a potential goal. By this, we can reduce the workload of drawing along root axes to selection of tip points, reducing the time for phenotyping a single image by several minutes. Lateral root counts can be estimated by the number of branch points along selected paths. Using these techniques, we have generated phenotypes for crown root number, the length of the main root axis, lateral root densities and lengths separated between L and S types, as well as S-type density on L types. For all of these traits, heritable genotypic variation was identified for 393 accessions grown under hydroponic conditions. We used a penalized linear regression model and publicly available genotypic data to establish genomic predictions of phenotypes for all accessions in the 3K RISE Genomes project. Prediction accuracies were estimated by cross-validation. We found non-zero prediction accuracies for all traits examined. As in stratified populations like the RISE gene pool, these estimates can be inflated by population structure. We looked at prediction accuracies within the major subpopulations Aus, Indica, and Japonica. While prediction accuracies for the same trait tend to vary between subpopulations, reasonable prediction accuracies can also be observed within each individual subpopulation for the majority of traits. We also estimated prediction accuracies of our genomic prediction models on an independent set of accessions grown under field conditions. I am here showing data for crown root number. We found that while overall prediction accuracies were reduced compared to cross-validation using only data from hydroponics, crown root number could still be predicted in the independent set of accessions with an accuracy of 0.52. Our work will enable us to characterize large sets of gene bank accessions for relevant root traits, making it possible to select donors for specific target environments and abiotic stress conditions. Once these models are established, predictions for root traits can be achieved from genotypic data alone, reducing the workload required for phenotyping rice plant genetic resources. With that, I want to thank you for your attention, as well as everyone involved in this work, especially our collaborators from Iowa State, Hirosaki University, and the team in Madagascar. I also want to thank my supervisor, Professor Visuva, and the PhenoRob Cluster of Excellence for giving me the opportunity to work on this.